Hi everyone, my name is Christina Hahn. I am the site coordinator for the Amgen Biotech Experience Program at the University of Toronto, Mississauga. We're here today just to show you a couple of components that are in our ABE program kits that go out to schools like yours and to teachers like you. So the first component of the kit that we're going to talk about is the setup guide. Now, we went through this fairly extensively in our training session, but just a reminder, the first page has a color code of where each consumable or reagent should go in terms of room temperature or fridge or freezer, followed by a list of all of the components and reagents that are included in the ABE kit itself. If you have notes, if something is broken, this is where you would actually report it. And then this is also followed by then a checklist that is broken down by lab and by locations for your ease. Just in case you're scrambling, this is where you're supposed to go to get all the information that's required. Each lab also has a setup page that includes pictures of what each station is supposed to look like, as well as arrows pointing out to the different components. Each lab also has a notes page as well that also shows you a couple of things and just reminds you of the things that are supposed to happen before and after each lab. As well, at the end there is a MSDS page, or the MSDS section that contains the MSDS for all the different components. All right, so in this next section, we're going to show you a couple of the boxes that are included in your kits. So you have room temperature boxes, the 4-degree or fridge boxes, as well as the frozen -20-degree boxes. And that's the one that we're going to start with. So this is your styrofoam frozen -20 reagent box. Please keep the components frozen. Either put this entire thing into your freezer once you get back to your school, or the Ziploc bag that contains all of your frozen reagents. Again, these reagents must be kept frozen, or else your lab 2 and 3 reactions might not work because the enzymes may have lost their viability or efficacy once they are thawed. This is your 4-degree box or your fridge box. This you will probably end up picking up when you pick up your competent cells. They contain your lab 5 plates, as well as either a school lunch bag like shown here, or another insulated box that contains your competent cells. That competent cell, these should go into your freezer, whereas the plates will stay in your fridge. Please keep these at those temperatures, or else your lab 5 transformations will likely not work. And last but not least, you have 4 or 5 of these Tupperware boxes, depending on your school size, class size, as well as your class number. Each box lid will also have a list of the components that are within each box, as well as any IDs of the equipment that ABE lends out to our schools, as well as the labs that each piece of equipment is used in. All right, so this is the first box of a set of 4 right now. This is the heaviest box that contains a lot of your big equipment. So to start off, this is the first piece of equipment. This is the transilluminator that's used to show glowing protein, either in the cells or in your fractions. Again, make sure that the orange filter is on top whenever the transilluminator is on, in order to protect you and your students' eyes from the blue transillumination light. These are your ice buckets, again, to put ice in for labs 2, 3, 4, and 5. Your lab 1 gels are already poured. These are for the dye running gels. They should be labeled dye running gels as well, just for your ease. You can also pour your own if you have Algaros gels from previous ABE kits, but I poured them for this specific kit. This is your heating block, so that's used for both the restriction enzyme lab 2 experiments, as well as for labs 3 and 5. And then last but not least in this section, or in this box, is the incubator that you use for incubation for all of the plates in lab 5, as well as the ice crusher that's just inside right now. Make sure for all of these big pieces of equipment that you unplug the power cord before you store them in there, or else the plug and the wires will become frayed and then I have to replace them. The second box contains the consumables and a bunch of other small pieces of equipment that is used for all the different labs. So first, starting off in the top layer, we have the student guide and the teacher's guide, both of which I will have a copy of each in every single kit that we send out. There's also online copies in our Dropbox unit, as well as I will usually email it to every teacher who has the kit. The benchtop diapers that are used for every station. Of course, you don't have to use these for your kids, but we recommend that just in case things spill or things are don't go as planned. The safety goggles that are used for this class specifically are provided if your school does not have them for chemistry or other classes for whatever reason. We do have them on site to send out with the kits. And then these are your main big storage Eppendorf tube racks, as well as the individual tube racks for each station. These are your P200 tips, and that is it for this layer. All right, for the next layer, first off, we have the Lab 1.1 practice gels. These are silicone molds of the gels that you will be using in Labs 1.2 as well as Lab 4. Again, the sizes of these agarose gels are the same as the ones that you're using in the other two labs, and they're just used for the students to practice before they actually get to loading the real gels. We also have the dye running buffers for Labs 1.2 and Lab 4, as well as some stock buffer solution that you will be using to make both the Lab 4 gels as well as run the Lab 4 gels with. In this box, you also have the gloves in small, medium, and large sizes, as well as markers that are used for all of the different lab components as well. And then in these little boxes or in Ziploc containers or bags are the room temperature reagents for Labs 1, 2, and 6. Again, they will be labeled on here in terms of what they are and how many copies or how many of each are in there. We also have the practice sheets for Labs 1.1, and these are for, in conjunction with the practice gels. They're just to help teach students how to pipette properly and accurately. Last but not least, we have the Sparkling powder solutions that are used for neutralizing the Lab 5 and Lab 6 bacterial waste, as well as the waste containers that everything goes into. Again, for the Sparkling powder, take 1 packet of each powder, put it into these containers, fill to the fill line with water, swish, make sure that is all dissolved, and that would be your neutralizing solution here. Right. Finally, you have the cell spreaders that are used for Lab 5, 2 different versions of it, depending on how many classes you have and how many people are in your class. And that's it for this box. All right, this next box contains a couple of the different smaller pieces of equipment that we will need to run Labs 1 to 5. Starting off with the blue gel units. These are used to run gels for Labs 1.2 for the dyes, as well as for Lab 4 to run your DNA samples. All right, so there's a couple of different components inside these blue gel units, and I'll show you what they are now. First, the power unit. Again, they have numbers on the power units that match the number that's on the bag itself. The blue gel unit itself, the gel running apparatus, it has an amber lid, the gel base, the reservoir where the gel will sit, as well as the bottom itself. There is a built-in blue light inside the blue gel unit. Please make sure that the lid is on before you turn on the light. Again, the amber filter is there to protect your eyes as well as your student's eyes. The blue gel lid fits only one way, with the blue gel on top and the minus on the top and the plus on the bottom. Next, we have the illuminator lid. This fits right on top of the blue gel unit itself once opened, and there is a little circle that the students can look through to see their DNA gels running. And finally, there is a Clearview screen spray, as well as a microfiber cloth. This can be sprayed onto the lid itself so that there's no condensation that forms on the lid, so that the students can see their DNA gels as they run the gel. Okay. All right, in this next layer, we have the sparkling squeeze bottle that's used in Lab 5 to neutralize and disinfect the plates that have been used in Lab 5 that have bacteria growing on them. So again, fill it to the top with water because there's already sparkling powder in here. Make sure it's dissolved and then just squeeze over top of the plates. To submerge the surface, leave for 48 hours and you should be able to throw the plates into the garbage. Next, we have the centrifuges that are used pretty much for every lab. Remember, there is always going to be a note to say, please either centrifuge the reagents before you use them in order to avoid losing them through the lid popping off while you're opening them. Make sure that the base is nice and stable, as well as there's no water or condensation on the lid. And when you pack up the centrifuge again, just make sure that the cord is unplugged and that you can pack it nice and cleanly. Finally, we have the P20 pipettes. Again, these are used in pretty much every lab in order to transfer small amounts of reagents from one place to another. Please do not turn them from the current volume to either below 1 microliter or above 20 microliters, as that will break the pipette and we will have to fix it. These are the agarose powder as well as gel green that's used for Lab 4. The gel green is used to visualize the DNA bands, so make sure that you do add that when you're making Lab 4 gels. And then finally, we have the Eppendorf tubes that are used for pretty much every single lab. Again, these are sterile Eppendorf tubes so long as the autoclave tape is sealing the lid. And these are packed either 1 per student group or 1 per 2 groups in 2 separate classes. All right, so if you're doing Lab 6, you'll also be getting a box that has all you need for the protein purification section. So first of all, you'll be getting a P1000 pipette tip box. This box contains tips that are used for volumes up to 1,000 microliters or 1 milliliter that is used with the P1000 pipettes. You will also be getting some HIC columns. These are the columns that are used to purify the proteins. Again, make sure that the columns are always stored upright, not horizontal, or else you will disturb the resin bed, at which time you won't be able to purify your proteins properly. These HIC columns will sit in a loaf pan or a waste container, and this container is just a loaf pan. It's very easy to use for all of the steps except for the collection of the red fluorescent protein. The column will be sitting in this pan. All right. For all of the different consumables and the reagents, they may come in a bag like this, or they may come in a bag like this, depending on the size of your class, as well as the number of students in each class and the number of classes that you have. All right. In order to use all of this, you of course need the pipettes that use the P1000 and P200 tips. So first of all, we have the P200 tips. These can be contained either in a Ziploc box or in a bag itself. And finally, we have the P1000 pipettes, which are here, and these are the ones that use the P1000 tips that I mentioned earlier. Again, make sure you don't move past or crank the pipettes past the minimum and the maximum, or else the pipettes will break. And that's it for Lab 6.